And we're live.
Great. Hello, everybody. Welcome to this special brown bag. For those of you who were here at the event last night, Buckley needs no introduction. But for those of you who took the evening off, I will give an introduction. So Rachel received her PhD from the University of Melbourne in Australia. I will not repeat the Florida joke. And has been working 8 years, I think it's 8 years, at Harvard University Medical Massachusetts General Hospital. She is currently an associate professor. I discovered not from your website.
Not because I've got to update that.
Associate professor of neurology, and her field is very well represented by the title of the talk today, which is the role of sex in differential vulnerability to Alzheimer's disease. We had a wonderful introduction to that field last night. She is a prolific publisher, I might say, and I, for one, am going to chase down quite a few of the more recent publications, given the interest of what we talked about last night. Without further ado, I hand the stage over to Rachel, and please join me in welcome.
Thank you, Tamik, and thanks to all of you for coming, especially those who came last night. There will be a lot of repeats, so we've got a lot of fun knowledge again. But I have really enjoyed my time coming here the last few days. I think that really the thing that's really struck me about coming to UTD especially is the amount of collegiality, the amount of connection between all the groups, and the passion and engagement of the grad students is so exciting. And that's particularly important to me and quite profound, because we don't have grad students at MGH. They don't exist. We do have Harvard programs, obviously, but it's very, very difficult when you're in a hospital environment to actually recruit the grad students to the labs there. So to see junior researchers just making their way through and not only clarifying their own science, but clarifying the science around them is really amazing. So thank you for giving me your time over the last while. And if anybody needs anything or would like to be put in touch with somebody, or if you'd like to see a template of a grant, my door is always open. My virtual door is always open. And if you ever wanted to visit, please do. Please look me up anytime. And I tend to go to a few conferences. I'm always at AAIC. I go to human amyloid imaging. So if I'm there and you see me, please come and say hi, or email me beforehand. So without further ado, I'm going to be talking a little bit about sex differences in AD. It's an area I'm super passionate about. I have sort of two tracks of research thinking about sex differences. One of them is really focusing on resilience versus resistance, and I didn't really touch upon that in this particular talk, although I should have, given the huge interest from this group. But it actually is interwoven in this talk, because I also am very interested in what are the sort of biological pathways that might drive sex differences, and I focus both on genes and hormones. And sometimes I like to pose the question, is it genes or hormones? But it always feels a bit patronizing, because we all know that it's a bit of both. But I'll start out a little bit first by giving a sort of fundamentals of where we've been for quite a while. So, you know, when we think about prevalence rates of Alzheimer's disease, dementia by sex, there is a much higher level of prevalence in women versus men for Alzheimer's disease dementia. Interestingly enough, what I didn't describe last night is actually that's very controversial, because when you think about incidence rates, which is the, let's say, where instead of just looking at a snapshot of society, you're actually counting how many people progress to the dementia within a life cycle of a study. Actually, the incidence rates between men and women are on par in the United States. Almost every single study that's ever studied that in the United States shows no real differences. Interestingly enough, though, when you go to Europe, there are quite a few studies in Europe that do show a difference between men and women. Why might that be? Well, there could be some sort of survivor bias related things. There could be some country related or recruitment related differences. But there certainly is some controversy in the field about to what extent are women actually progressing to the dementia more than men. But what one can say, and we do see that women show greater rates of mortality with dementia, but what we can say with quite a lot of confidence is that women who carry E4 are at greater risk of progressing to the dementia. And that is something that is far less debated. There are greater incidence rates to dementia if women carry E4. This is one example. This is one of the biggest and most recent examples of these studies, but there have been many of these studies conducted over decades in multiple sort of really large epidemiological cohorts that had some measure of E4. And this is one done by Judy Parr and a few other colleagues, a few years ago now, where they combined 23 data sets across the GAIN platform. If anybody's familiar with the GAIN platform, it's a freely accessible online platform. And what they found was that women who were E4 carriers had higher rates of incidence, but only within a certain age frame, which is quite interesting to me, and how I have described it in many of my grants, is that this age range of 65 to 75 is sort of an early age, earlier age than what we would normally deem for incidence rates for dementia. But if it's critical to show that, if it's a critical time frame for sex differences, one might imagine there might be something 20 years prior that's having a real influence on that. And one of the areas that we're really interested in that might be 20 years prior to that age range is around the time of menopause. So what is the data that's beyond dementia? We talk a lot about dementia risk, but actually what I'm more interested in is the pathology. What are the sort of evidence to sort of describe sex differences in the pathology itself? And this is one example from a couple of different, studies that came out around the same time. This is from the FLAME autopsy cohort, but there was also a similar study, Ovisgaran. I actually don't know how to say his last name, but that was also based in the ROSMAP autopsy data set. There were two very similar studies that came out showing that women and men do not have very many differences in amyloid plaques in the brain, but when they start to look at neurofibrillary tau tangles, that's where you see much greater levels of tau tangles in women relative to men. But this is particularly focused in sort of medial temporal regions, primarily in the hippocampal formation, and in certain ages. They were primarily finding differences between the ages of 50 and 70 in this particular work. Now, what does this sort of translate to? We see in a lot of our work that actually women are quite resilient. So for a given level of pathology, women tend to actually perform a lot better than men cognitively. And this is sort of where the argument of are women susceptible or are they resilient comes in. Because in a lot of the studies of autopsy data, they've often said, well, women have higher levels of neurofibrillary tangles. And this was Lisa Barnes' study in the ROSMAP cohort from 2005. They suggested, well, for a given level, if you equate men and women on their level of pathology, and they both have very severe levels of pathology, actually women are performing worse just before death. Right? So there was a lot of feelings that early on, there was a lot of arguments about vulnerability and susceptibility and frailty in women. But actually, there's another very prevailing idea now, and Jessica Caldwell has been one to really drive this from the Cleveland Clinic, suggesting that women actually perform a lot better than men on a lot of different cognitive tests. And they do so for a fair period of time. And then suddenly, or at some point, there'll be a great hinge point and they will start to decline rapidly. And this is one example of those. So the red line here, so the essential argument of what I wanted to look at is for a given amount of amyloid and E4 carrying risk, do women and men show differences in their rate of cognitive decline? And what we found at the intercept is women who had high levels of amyloid and were E4 carriers blitzed men on any composite I could think of any cognitive composite. Here it was just a global cognitive composite, but they outperformed men who were high amyloid and E4 carriers. But what we found was women in that red line had way faster rates of decline than the men who were in the green line. And this was some of the first evidence that I really felt quite interested in to really make me sit up and take notice and think, maybe it's not really an argument for susceptibility and vulnerability necessarily. It might be more of a case of women showing a lot of resilience and then at some point losing that over time. But at that point, you know, we've been doing a lot with amyloid PET, and we've been trying to see whether there were sex differences in amyloid PET. And at that time, we had about 900 scans. We've now included A4 data at the baseline. So now we have 4,400 scans. And across all of those, you know, thousands and thousands of scans, men and women do not differ in their level of amyloid, at least not when they're older. I'll put that little asterisk there. At least not when they're older. Men and women do not seem to show a difference. But where we were starting to see, and what replicates nicely with the autopsy data, is that in tau PET, here what we've got, what I'm showing you is both Harvard Aging Brain Study participants as well as ADNI participants. So we've harmonized all the data. We've put everything through the same sort of neuroimaging pipelines. And we're simply comparing at each vertex, what is the difference in the amount of PET signal between men and women after adjusting for age and cohort. And what you can see here is not just limited to the medial temporal lobe. You're seeing sex differences in tau PET signal across the cortex. There's a lot going on here. And you certainly see in this little tiny, you know, area here where you'd imagine there to be the entorhinal and the sort of medial temporal regions, there's certainly this little hot spot that's that's showing the sex difference. But we actually characteristically see a lot of areas of the brain. Interestingly, something that comes out over and over and over again is the lateral occipital region. And but we see, you know, middle frontal region, we see parietal regions, we see a whole bunch of different regions that show sex differences. One of the things that we were concerned by, because we're really big PET people, really concerned about the artifact from tau traces. One of the problems that we have with tau PET is that it has a lot of off-target binding. There's also issues with, you know, partial volume correction, how you deal with, you know, morphological brain differences between men and women, right? So all of these different things, we've actually worked a lot for many years trying to dig into a lot of this. And some of this can't definitively be answered. But we published a paper recently looking into some of these really problematic effects. And we see massive sex differences in every one of these things. There is off-target binding. There's more off-target binding for women. We also see some issues with partial volume correction. So there are partial volume effects that differ between men and women. And obviously morphological differences play a role as well. So brain volume differences and so on. But even after adjusting for some of these things and equating for some of them, we still see massive sex differences still appearing in a lot of these regions. Some of them do attenuate, but a lot of them remain the same. The other interesting thing about this is we've managed to repeat a lot of these findings across different traces, across diagnostic status, and in different independent cohorts. So it's you know, a lot of these regions are quite fascinating and deserve looking into. So what is this high level of tau, you know, impact? So before, when I was showing you these longitudinal changes, amyloid and E4 were really driving faster rates of cognitive decline. But one thing we know for sure is that tau is extremely proximal to cognitive decline. And so what we wanted to see was, does tau have an impact on rates of cognitive decline in women over and above what we might think of as the effects from amyloid and E4? And indeed, after adjusting for amyloid and E4 in these models, we see that tau has a very, very big impact on rates of change in women. And in this particular analysis, what we've done is we have not equated men and women on their level of tau. This is just simply a visualization of different tertiles of tau and what types of rates of change you might expect. But if you do equate women and men on their levels of tau and see how fast the rates of change are happening, women are still showing a faster rate of decline. But one issue that comes out in my mind here, one big caveat that I've always worried about, and I cannot necessarily easily get around it, although there are some statistical ways you can try to simulate these issues. But we don't know to what extent there's a survivor bias here. One thing that really worries me is that those men look extremely healthy for a very, very high level of tau. And we know that traditionally people don't do well holding on to anything when they've got that amount of tau, which makes me interested in how many of the men who have high tau already have already progressed to a diagnosis or are not, like, cannot be studied for whatever reason. And so this again is another example of, is what I'm showing you here a marker of susceptibility, or is it a marker of resilience? So these women were healthy at the outset of the study and could continue to be studied. So we've shown you, a little bit of autopsy data, which has to be by definition cross-sectional. And I've shown you some cross-section data of differences in tau. But we do have issues with survivor bias when you look at cross-sectional data. One thing we're also interested in is, can we actually see changes in tau as well? Is there faster rates of tau accumulation in women relative to men? And are there moderating effects on this? And my incredible postdoc, Dr. Gillian Coughlin, she's actually not a postdoc now, I keep saying this, but she's now junior faculty starting from early next year, which is very exciting. So what she's looking at is trying to ascertain the extent to which women have faster rates of change on a tau radio tracer across multiple different cohorts. So on the very left here, you've got ADNI and Mayo, the Mayo Clinic Study of Aging. You've got Biofinder 1 at the top there in the middle, and then the Wisconsin Registry for Alzheimer's Prevention, and on the very end is the Harvard Aging Brain Study. This is something that we do constantly in our lab. So we will not just use one data set. We will often look across multiple data sets and get an aggregate of what the finding might be. Because I've never been able to live with myself doing one and done and just believing it's true. And what you can see here is overall there's a trend. But it does seem to be moderated by amyloid. So in high levels of amyloid, women show much faster rates of tau accumulation than men. Now, that's not true of every single study. You can see that there's some nuances there, and certainly there seems to be an impact at the intersect between men and women, and certainly true in some studies relative to others. But across the board, there seems to be changes. So what does this sort of lead to? This leads to a working hypothesis, at least in our lab, which is, in our hands, we don't really believe that there necessarily is a difference in the amount of amyloid, or there doesn't seem to be an influence of sex on amyloid accumulation or amyloid at the cross-section. But once you hit a level of abnormality in your amyloid, this is when it starts to bother the tau. And rates of tau accumulation and also cross-sectional levels of tau between men and women really start to differ. This then seems to have a knock-on effect for rates of cognitive decline, which may then impact higher incidence rates of dementia, although that is somewhat debated. We don't have that sort of data available to us, unfortunately, because incidence rates to dementia and diagnosis in a lot of our studies is really hard to come by when you're starting out healthy at the beginning of a study. We'd have to measure people for a long, long time. So I'm going to now switch tracks now that I've given a bit of the background of where we had been, and now I'm going to talk a bit about where we're headed. So I want to talk a little bit about the hormones versus genes component. So menopause is a really, really life-changing event in a woman's life where you have a massive cessation of two really important hormones, estradiol, as well as estrone and progestin. And then you also have, in a man's life, a slow decline, an insidious decline of testosterone. In some cases, they call that andropause, but that's sort of a controversial term, actually, because it is such a slow decline. There's not an actual precipitous drop. Some men do have that for certain medical reasons, but in all, it's not really the same as menopause. So they're a little careful with that. But what you can see here on the right-hand side is I wanted to sort of describe another element here, which is the sex hormone binding globulin. So as people decline in their levels of testosterone, for instance, as shown here, one thing that does increase over time is sex hormone binding globulin, which is a glycoprotein that is ostensibly its primary function is supposed to be involving the bioavailability of sex hormones. It helps to transport sex hormones to where they need to go. And so when you have declining levels of sex hormones, there's more sex hormone binding globulin just wafting around doing nothing. But that is not actually its only function. A lot of people would think that, but if you're really sort of deep in the literature, there is actually also a lot of functionality of SHBG with vascular related function as well. But we'll come back to the hormones in a second. I want to start out by just talking at a very broad level about menopause status. There have been a few pieces of data coming out of Lisa Mosconi's lab looking at menopause status and Alzheimer's disease neuroimaging markers. I do want to preface these findings with the fact that a lot of these data are not massive samples. So their first work was only in 16 people in each group. They have increased it a little bit, but I just want to provide that caveat. The other caveat to a lot of the work that looks at women going through menopause is that, and you guys would be very familiar with this because of all the PET imaging that you do, but these are women who are traditionally between 40 and 55 years of age. And I know these guys, you guys would know better than anybody looking at sort of earlier life stages and PET imaging, but there's a lot of signal to noise issues here. So we've just got to bear a lot of these things in mind, signal to noise ratio and sample size. But bearing those sort of caveats in mind, Lisa found some really interesting evidence to suggest that, as you would expect, going from asymptomatic perimenopause, which is quite young in a woman's life, through to perimenopause, which is considered to be a fluctuating stage where you have, very irregular periods. You may be starting to get quite a few symptoms of menopause, but your periods haven't stopped for longer than a year. And then finally, menopausal women, which is considered to be periods stopping for at least a year. And throughout these stages, already you can tell that there's going to be very big age differences, right? So bear that in mind. But they see increasing levels of amyloid. They see lower gray matter volume and lower white matter volume across these stages. But the other thing they did as well is they compared each of these groups to an age-matched male group that was equal in sample size. And what they found, even against those age-matched males, and you can debate with me all you like, because I also agree with this, I struggle with this myself, are men a good reference? Who's to know? But against those age-matched males, they find higher levels of amyloid at each of those, at the perimenopause and the menopause stage as well, and lower levels of grey matter. So we found this kind of interesting data. We took a slightly different approach. And we went to the Framingham Heart Study, where they have just, started to, well, not just started, I mean, in the last 4 or 5 years now, have been doing amyloid and tau scanning, quite a few adults who are now probably 50 to 70. So they're a bit younger, but they're not as young as the Mosconi sample. And they have a lot of really good phenotyping of menopause, actually. And what we wanted to do is sort of try to replicate what was being done in the Mosconi group, but extend it to tau PET as well. And interestingly, we saw some weak signal with amyloid. There were some hints that maybe postmenopausal women showed higher levels of amyloid, but it didn't really survive correction for multiple comparisons. Where we tended to see quite consistent and repeatable effects were in regions of the brain, looking at tau PET signal in regions of the brain that seem to show, how I pointed out some regions across the cortex. These are regions we will tend to look at now when we're looking at sex effects. So things like, you know, the entorhinal, the inferior temporal, lateral occipital, middle frontal, inferior and superior parietal. All of those regions we will look at specifically to see whether there are differences. And indeed, in age-matched men who were postmenopausal, there were lower levels of tau signal in almost every region of the brain relative to postmenopausal women. And postmenopausal women also showed higher levels of tau relative to premenopausal, although that was a relatively weak effect because there's lots of variability here. But one thing that we did find really fascinating was when we started to turn to a different cohort again. So this cohort now, this is Dr. Coughlin's work, is looking at the Wisconsin Registry for Alzheimer's Prevention. We work with a lot of different data sets for various reasons. One is because each data set has its own limitations. When we want to look at reproductive history, almost every single cohort has either not gathered that information or it hasn't done it in a very consistent way. So there are only certain things we might be able to ask. But here with the Wisconsin Registry for Alzheimer's Prevention, they have actually a really beautiful curation of reproductive health. So if you're ever interested in it, I would highly recommend reaching out to that group. But they have quite a large collection of amyloid and tau data. The one amazing thing about this tau data is they get MK6240, and that Merck tracer is far better than flutalsapir, I'll give you that much. So you get much better resolution, stronger signal. So what we were looking at first and foremost is the moderating effect. So we know that amyloid and tau are quite closely correlated, and we wanted to see whether any of these reproductive factors of age at menopause, hormone therapy use, or in fact, the time difference between age at menopause and hormone therapy use might moderate the association between amyloid and tau. Oh, there's a public safety message going on. Okay. But what we found was that earlier age at menopause was associated with stronger associations between amyloid and tau. The argument there we were thinking was that earlier age at menopause means you are having less exposure to estrogen throughout life. And that can actually be considered across many human and animal studies to be less protective. And so that sort of fit in line with the literature and what we would be expecting. But what really confused us at the beginning was that we then thought, well, hormone therapy you should technically, because you're reintroducing exogenous hormones into the circulating system, therefore you should be reintroducing protection. And they should have a less association between amyloid and tau. And in fact, we found the opposite. Women who reported using hormone therapy at some point in their life, so these women were a bit older, these women were on average about 65, so they would have gone through menopause a while ago. But we found that hormone therapy use was associated with higher levels of tau and amyloid. And so Gillian decided, okay, well, that's not the end of the story. There's clearly got to be something that might drive that. And what she did then was look at this amount of time since they initiated hormone therapy use. And the argument behind that is because there's actually been a lot of evidence since the Women's Health Initiative data came out. That was a massive clinical trial that failed a couple of decades ago, looking at hormone therapy on risk of a lot of different outcomes. Dementia was only a secondary outcome. They were actually interested in things like endometriosis, heart health, osteoporosis, heart health, and a few other indicators. They were not at all interested in dementia, really. But they looked at it as a secondary outcome and found, in amongst a whole bunch of other things that were going wrong, that dementia was twice as likely in women who were taking a combined therapy of conjugated equine estrogen and progestin. And so one of the big sort of fail points that people came away with was, well, why would you give women who are over 65 hormone therapy when they went through menopause at the age of 50? It makes no sense. You've like had this massive gap of nothing. And then you suddenly reintroduce an entirely potent neurogenesis factor, which caused rampant cancer risk as well in those women. So, so there's now this sort of theory called the window of opportunity, the ideal time in which you can reintroduce estrogen without causing the horse to bolt, so to speak. And what they found was, what we found in our data was very in line with that. So this is a visualization of groups. We didn't do that. We used a continuous variable, but essentially we created a variable of how long in time between initiating hormone therapy and menopause onset. And we found that further delay was associated with greater risk. Now, why would you delay? Who would actually delay hormone therapy? If you're going through symptoms, surely you'd be asking to have an amelioration of those symptoms. There are cases, it is more rare for women to delay, but there are cases in which women will need to delay for various health-related issues before they can initiate hormone therapy. At least that was the case prior to the Women's Health Initiative data coming out. It is very rare now for women to do this. Most women, if they are recommended to initiate hormone therapy, it will be around the time of menopause. And it's because of data like this that shows that a delay is not a great thing. But what about sex hormones? So I talked about sex hormones very briefly early on. And we've been really interested in trying to understand whether endogenous circulating levels of hormones in the blood might have any indication of levels of AD phenotypes that we're interested in. In this case, we're usually interested in amyloid and tau. And this is a study that was conducted by Erin Sunderman and colleagues on the ADNI data set. ADNI does have some hormonal information, interestingly. They have FSH levels, which have not really borne out a huge amount. I think there were some issues with their panel. But they also have testosterone. They were not able to gather estradiol, unfortunately, but they do have testosterone levels. And what Erin and Sarah Banks did, based at UCSD, was they wanted to look at how testosterone levels in the blood were associated with cerebrospinal fluid levels of p-tau, phospho-tau 181. And what they found was that there was an interaction between with E4, suggesting that E4 carriers actually showed a stronger association between testosterone levels and p-tau. But the really interesting thing, I just want to apologize to everyone. I don't know why they did this, but they changed the colors here so that like the E4 carriers are green. These are still E4 carriers, so don't get confused. So what they found was that the driving association here was not in men, which was kind of confusing. What they found was, and they actually standardized the testosterone levels so that they're similar between men and women, which is a little bit confusing, but they did it nonetheless. And what they found was that in women who are E4 carriers, that was where they were seeing the strongest effect. So there seems to be, at least in their hands, some kind of association between testosterone and phenotypes of interest in AD, at least in women who are E4 carriers. They didn't see a main effect of sex just through E4. We then decided, okay, we actually have testosterone data. We also have estradiol levels and estrone levels and SHBG in Framingham. So why can't we do a similar thing but look at it in relation to amyloid and tau PET? And so that's what we did. Now, interestingly, we found no testosterone associations. We weren't able to replicate the findings from ADNI. That's not to say that those findings don't exist, and I think they are very interesting. But we, in our data, were not able to replicate that. But what we did see over and over and over again was SHBG associations. So we found higher levels of SHBG to be protective, at least in men. So that was very interesting. We found no effects of testosterone. We found no effects of estradiol or estrone in association with amyloid or tau PET. But we found sex hormone binding globulin, at least in men, was associated with being protective against higher levels of tau. And this was particularly true when you moderated by E4 levels, although just trend level. And it's not trying to figure out what the interpretation of these data are, but if SHBG is somewhat associated with vascular function, which is what we are now testing, because we've got some vascular neuroimaging markers and some angiogenesis measures in blood, We want to test whether SHBG is having some kind of ameliorating effect on some kind of vascular function, which may be having a good outcome for tau PET signal, but remains to be seen. So I want to now turn to the X chromosome, because this is an area that I'm really super passionate about, and I think it's an area that's deeply underexplored, and I think people are now starting to really look into this. So the people in this space that I would really read their work is Dr. Dina Jubal, based at UCSF, and Dr. Michael Belloy at WashU. So I don't know if anybody is interested in the X chromosome at the moment, but Dr. Belloy just put out one of the biggest XWASs out there, an XWAS of AD, and it's fascinating. And they found one very interesting novel hit, which requires further exploration. But this field is starting to expand. The reason why people have not looked at the X is because it's very technically difficult to do, but if we can do it, it is worthwhile. And why are we interested in the X chromosome? Well, the women across the globe, doesn't matter where you are, they will live longer than men. And in fact, women also live longer with dementia as well. So there's really another argument for resiliency here. But is it about the ovaries particularly, or is it something to do with this XX chromosomal element? And one of the strongest argument, particularly from evolutionary geneticists, is that it's to do with the XX. And you can tell it immediately on the left-hand side here. This is a beautiful depiction. I've stolen this from Dina, so huge shout out to her. But this is just a beautiful depiction of the longevity, the lifespan of different species, based on whether they are homogametic or heterogametic. And heterogametic means you've got two different sex chromosomes, and homogametic means you've got a repeat. And you can have across species, there are examples of female heterogametes and male heterogametes. And obviously we have male heterogametes in the human species, so they're male XY. And these are all represented by the blue dots here. So male heterogametes tend to die earlier, their lifespan is shorter. But you can see there's actually a lot of species like birds, some moths, some fish even, and lizards and things like that, that are female heterogametes. And you can see that those also tend to die off earlier. They have ovaries, but they're dying off earlier. And one of the strong arguments there is that there's something to do with this XX that allows you to have this wonderful compensatory mechanism. And one thing that Dina decided to do, which I think is absolutely amazing, is she took this idea of the XX being special, and she went back into the literature. Art Arnold, who I believe is based at USC, but I could be incorrect there, he developed something called the 4-core genotype model, where you're able to take the SRY. So the SRY gene is the thing that turns on or off testes. And you can actually manipulate that. You can take it off where it's supposed to be, put it on the autosome, and you can flick it on and off at will in a mouse. And by doing that, you can actually make male gonads develop or not. And you can do that in an XX and an XY being. So you can end up with what we would expect naturally, so a male XY and a female XX, but you can also end up with the cross example, which is a male XX and a female XY. So Dina decided, okay, so we've got this 4-core genotype model, and I want to see if the X chromosome is having more of an impact than, or what's the impact of gonads versus the X chromosome on Alzheimer's disease. So she simply said to herself, maybe I should take this 4-core genotype model and pair it with an AD mouse model. It's such a simple idea, but so smart. And what she wanted to do with this 4-core genotype model is actually split apart in an experimental way which component, is it the gonads or is it the sex chromosomes that's having an impact on disease progression? And here she was simply interested in survival. What she found is not surprising to anybody, but knowing both gonads and genes have an impact on survival. They have the biggest impact together on survival, and it's males that tend to, it's the XY females that are doing really poorly. So something about, the two together. But the one thing that really struck her and me was that the sex chromosomes out of the two seemed to have a much stronger impact. But she didn't just stop there. She didn't just stop, because here you can't really tell, well, is it the Y that's the problem? Or is it something about a loss of the X? Because you could say that, well, the Y is a really piddly little chromosome. There's not many genes on it, and they don't have a huge amount of functionality, unfortunately. So the Y is sort of this weird lingering appendix or something. Whereas the X's, they're really sort of quite fabulous, and they are very powerful sex chromosomes. If you're just getting one from your mother as a male and there's a mutation on it, you could be in big trouble. So what they wanted to know, is it something about the Y or is it something about the X? And so what they did was they created another design where they were pairing the XX and XY together to create 4 different groups here, where you had sort of the XX, the normal XX example, you had an XXY, which is like Klinefelter, you had an XY, which is sort of normal male, or you had an X with nothing else. And the animal that had the poorest survival was the X with nothing else. And what they took away from that, and even more so than anything to do with a Y, and what they took away from that is it's a loss of X in particular that's the issue. So why are we so interested in a loss of the X? What could that be doing? Well, in women who have 2 X's in their body, in order to avoid overdosing, every single cell of the body has one of the X's randomly silenced. And that happens in a very, very random way. And so we end up with this beautiful mosaicism. Does anybody know what a calico cat is? So the calico cats are beautiful. They've got these patches that are all over them. They're actually an example of random X inactivation. They only occur in the female cat because of this random X inactivation. What is so incredible about this inactivation process is it's imperfect by design. There are X's that leak out, and every single woman in this room will have a different level of X inactivation, that escapism. And the really fascinating thing, there's some really amazing researchers, basic scientists who look at this. Dr. Jeannie Lee at the Brigham is one example in Boston. And what she said to me the other day is, we've looked at the amount of escapism in animals. Those animals, the female rodents that have less escapism, they're more male-like in features, which is really fascinating. So there is definitely a huge amount of variability across the population, but there are some genes that tend to characteristically escape inactivation across the population. There's about 240 of them or so. And so what we thought we would do is look at gene expression on those genes that tend to escape inactivation across the population and see, might these genes be associated with phenotypes of interest? The other thing that's kind of interesting about a lot of these genes is that they're involved in immune pathways and inflammation. And we know that inflammation and immunity is very, very heavily tied into Alzheimer's disease. So we thought we were kind of onto something here. And so Dr. Mabel Seto is one of the postdocs in my lab. She's absolutely fabulous. She came from Vanderbilt. And we do a lot of work with Vanderbilt, Dr. Timothy Homan and Logan Dumitrescu. And what we found was, out of all of these genes, we did find 3 XCI, so X inactivation genes that tend to escape, that were associated with outcomes of interest. So this is one example, CNKSR2, which is a connector enhancer of kinase suppressor, and a scaffolding protein, and it's been associated with X-linked intellectual disability. And what we found was that in only women, in female stratified models, women showed an association of higher levels of amyloid with lower levels of this gene expression, suggesting that it might be protective. So higher levels is lower levels of amyloid. And this was not true in males. There was no association. We also found an association for tau tangles, but the opposite direction. So higher levels of expression was associated with higher levels of tangles. We found some other hits as well. These were the other two hits. The reason why we're so interested in these two hits, JPX and FTX, these are some of the biggest mechanisms, the functional mechanisms associated with exon activation. So these guys help exist. Exist is the big sort of random classifier that turns off or on your maternal or paternal X in each cell. And these JPX and FTX are inherently associated with how well the exist works. And what we found was that these genes were very, very strongly associated in sex stratified models and also in interaction models with both tau tangles and cognition. So we do think there's some interesting associations coming out on X-linked genes. But the thing that was really fascinating to us, this is what we're showing here is a series of volcano plots. And what you see in each of these plots, this is every single one of these dots is basically a model of how much does this gene predict that outcome after adjusting for some covariates. And what you can see in this direction is this is the estimate down here, and this is the p-value. So if it's going in this direction, it's suggesting there's resistance. So a higher level of gene expression is lower levels of amyloid, for instance. Higher level of gene expression is associated with more amyloid. And so what you can see is there's both risk and resistance coming out. And what we've also done for each of one of these panels is we indicated whether it's a male-specific association that's significant or a female-specific association that's significant. Anything above this line is a significant effect. And what you can immediately see here is there's a lot of red dots. There's a lot of red dots. Is this surprising to us? Probably not. But it's really interesting. This is not just, and also just to highlight here, this is not just the X chromosome. This is the X and the autosome together. But you can see there's a lot of things going in just females to suggest that gene expression pathways are associated with phenotypes of interest. And in fact, what I would go so far as to say is that there is a lot of argument for why there might be very sex-specific biological pathways that are sort of very sex-specific to get them towards dementia. But I have focused quite a lot on the X, and I do want to give a shout out to the autosome because we have sort of broadened. So with our collaboration with Dr. Dumitrescu and others in her lab at Vanderbilt, we've been interested in gene expression across the autosome and how gene expression across the autosome might be associated with endophenotypes of Alzheimer's disease. But what we're interested in is not the ones that are, similar or similar across men and women, but what you can stratify. What is actually very different by male and female. And what you can see in the circus plot here, the F, the P, and the C in green, these are all the associations with outcomes of interest by gene expression in different regions of the brain. So this is the frontal, this is the parietal, and this is the caudate. And each of these regions are showing a lot of associations with outcomes, and these are the same by sex. Okay, so these are associations that have been published before. And just as a quick shout out, this is ROSMAP data. What we found here in these analyses, though, are sex stratified analyses. So all these sort of pinky orangey associations are just in women. So these are the associations, gene expression in the frontal region and brain tissue from the frontal region, from the parietal region and the caudate that are associated with amyloid, tau tangles, and longitudinal cognition in ROSMAP. And you can see in this tiny little sliver here, these are all the male specific. And what we found over and above was that women there were many more female specific associations with outcomes of interest than men. And just as an example, some of these ones that they pulled out here that they thought was interesting was PTRHD1 and CCL28 that showed sort of, I think what I take away from all of this is that there's sort of female vulnerability and female resistance, male vulnerability and male resistance on a lot of different genes. But some of these are actually very brain relevant and very associated with pathways that we would think are relevant for Alzheimer's disease. So I wanted to sort of end off by saying that obviously it's both hormones and genes. I think that there are direct pathways getting us from genes to outcome, but they are also very likely to be going through a huge amount of modifiable and other sort of biological mechanisms to get to that point. The same is true of menopause. So let's say, in menopause, so many things change, not just estrogen levels, or by definition, as a factor of changing estrogen levels, you get an increased risk of metabolic syndrome and overweight. You get disrupted sleep, poor mood, cardiovascular risk goes through the roof, and hormone therapy use comes online. In addition, on the side of sex chromosomes, you get there's a lot of very relevant immune system and neuroinflammation pathways that are implicated by escaping genes on the X chromosome. And all of these combined is what we're looking to target to understand how women might be at greater risk of AD pathology. But there still remains a lot of questions that are unexplored. One of the biggest ones is trying to figure out what is the impact of survivor bias. The men that tend to be able to go into a lot of the studies that we have, ADNI is a perfect example, but Harvard Aging Brain Study is another one, where in order to get into the study, you have to be very clean. No previous history of stroke, no previous, major cardiovascular illness. The list goes on. And in order to achieve all of those eligibility criteria, I think that does cut out quite a few men. So we do end up with interesting survivor bias. It's something that's very, very hard to try to quantify. In addition, there's a lot of questions to me about why women with E4 tend to be at greater risk. What is it? What could be the pathway there? And I think there are some researchers out there really doing a lot of this justice. Dr. Roberta Brinton has done a lot of this work, and Lisa Galea. But still more needs to be explored. What is the sex-moderated role of inflammation? We don't know that, but we know that women have a very different inflammatory profile than men. And because inflammation is so heavily implicated in AD, it'd be crazy to ignore this. The question of whether females are resilient or susceptible is another good question. I think you can argue for both, but I'm starting to really err on the side of resilience now. What is the added level of gender? I tend to ignore gender in a lot of my modeling, which is very bad, because it does have a huge implication. So women in these birth cohorts originally had different levels of education to men. They had different occupational attainment to men. They may have had different access to quality healthcare. So a lot of different things that differentiate men and women, and certainly also by race and ethnicity. And a lot of this work has been underexplored and needs further work. There's also something else I've neglected to look at myself, but is really necessary, is to look at gender identity. So there's a lot more work coming out now in the LGBTQI space, and we really need to sort of spend a lot more time understanding what it means to have that added level of complexity to gender and gender identity on risk for Alzheimer's disease. So to finish off, I just want to say thank you very much to all of my collaborators and colleagues, and they are friends, to be honest, and the Harvard Aging Brain Study Lab, where I do all of my work. My funding sources, for sure, both philanthropic and national funding, as well as my girls, who are absolutely awesome. This is our trip that we did just recently through Utah, and what an experience. I wish I'd done that at the age of 5. Thank you very much.
Very much, Rachel. Fantastic. We're open for questions. I think you were a microsecond quicker.
It's very quick. So what is the prevalence of APOE, and is there any difference between the prevalence in men and women?
By definition, there isn't, no. But just extending on that a little bit. So the prevalence of APOE in the studies that we tend to see, it's about 26%, but that's not the real population prevalence of E4. If you're thinking about homozygotes, it's probably less than 1.5%. In the general population, the estimates vary from about 15 to 25%. But just to extend a little bit, something we're really interested in is, where did your E4 come from? Did it come from the mum or the dad? And so we're sort of really interested in genomic imprinting right now, and really getting to the bottom of that. Problem is, you don't normally have generations of E4 status. The only place we really know that exists is in the Framingham study. So we just pitched that idea to them and they loved it. So hopefully we'll be able to have some data. So watch this space. Yeah.
Karen,
I'm curious what you know about, is there a plausible mechanistic link that's been hypothesized? I know it wasn't replicated, but this testosterone to tau association, is there like a plausible biological pathway that people are interpreting and discussing?
Yeah, not at this stage. So they said in the paper actually, so I was curious myself what exactly they were assuming might be a potential mechanism. I'll send you the paper. It's a really nicely written paper. They had some pathways that they suggested, but without, I mean, there's not really a huge amount in ADNI that would allow them to test some of these questions so clearly. But they did point to some animal model studies. So I think I'll have to send you that paper so you can have a look.
So I'm curious, are there, say, normative levels for what testosterone should look like in people? It would be age, right, bands, right? So
Yes, I think there are, although in women already they're very suppressed levels. So I think what they had to do to really look at this, look at testosterone levels in women was a little bit fancy, how they sort of tried to normalize the data or standardize the data and then look at the outcome. So I'm a little curious about how exactly they ran everything. But yeah, in terms of knowing normative levels in men, there's quite a lot of data out there.
I know I
For
can
women,
read the
not
paper
funded.
too, but also was estradiol level controlled for?
They
It's
couldn't,
showing
they
that effect?
couldn't, unfortunately, because there
Okay.
wasn't estradiol collected.
Okay.
One thing that's kind of interesting, and this is anecdotal, but I've spoken to a lot of menopause doctors now, and quite a few of them recommend giving testosterone to women.
Yeah, and I'm wondering like what's the scientific base?
So there has actually been not any data that I've seen on testosterone being given to women, but there have been a lot of clinical trials of testosterone being given to men in later life to see whether that can impact cognitive change in men and be protective. Data is mixed at best, and there's no clear effect.
Thank you.
Yeah, great question.
just had a question on the GWAS data. So I know you know this very well, but just to make it clear, that a significant effect is not the same as a high penetrance.
No.
And so all of these genes that you were showing that have what are called hits
Yes.
means that obviously there's a greater than chance statistical association
Exactly.
within some kind of variable.
Yeah.
But the amount of variance that any one of those genes is accounting for is, if it's anything like things like schizophrenia or depression, it's fractions of a percent.
Oh, absolutely.
So given that, you know, the same issue that comes up with things like schizophrenia, whatever, you have like hundreds of genes which all confer a tiny bit of vulnerability, whatever. How do you use that data to think about what really is driving risk that we can in some way intervene and manage in respect of Alzheimer's disease?
So when it comes to sort of very low effect common genes, right? Those, in my mind, the way I think of it, when we see hits like that, the first thing that you really must do is try to replicate that finding in any way you possibly can. Validation is huge, but it's not necessarily for drug prioritization. So I wouldn't necessarily take those collection of hits and say, okay, we're going to see whether those drug targets are going to be therapeutic. But what it can tell us is more about biological pathways that could be of interest that we could maybe manipulate in mouse models or try to understand a little bit further about what the biological pathways and the mechanisms. Specifically, what I'm interested in is trying to determine to what extent we have shared pathways to get to dementia and in what ways we don't, to try to optimize personalized medicine approaches. But the other thing that we're sort of interested in now, which is really hard to do, and I'm not sure it's going to pay off, but is to look at rare variants, because that's really where you have the biggest power, right? If you can find a rare, so for instance, what we're really interested in right now is trying to figure out rare variants of resilience or resistance to disease. Because if we can find that, something like HIV, they found a really penetrant rare variant that then changed the game, and they were able to drug that and, you know, change the outcome of HIV treatment. That's sort of where I would be going if I'm thinking about actually changing disease course. But when you're thinking about these sort of more common variants, it's more trying to, in my mind, understand what might be relevant, brain relevant, that can help us understand sort of biological pathways. But to be fair, a lot of these hits don't necessarily replicate. We find a lot of trouble with that, and especially when you get to these sex-specific levels, these are very weak effects. So even though, you know, we talk about, and let's be completely frank, if we're talking about hormone effects and sex chromosomal effects, these are still weak effects relative to the big hitters like age, E4, you know, against major risk factors of Alzheimer's disease. These guys are, you know, but you could say that of a lot of the things we study, but it still can give us really valuable information about how things are working in the background and what we might be able to better glean for changing the course of the trajectory in the future. But in and of itself may not be the deciding factor. Yeah. Oh, Ezra.
Yes.
Yes.
About the mouse models.
Yes.
My understanding is that mice don't go through the pause. So is that like how much does that limit their utility? Or do you think it's
Yeah, they don't. You know what's really interesting is they've now developed a really amazing mouse model. So Dr. Kristen Zuluaga, who's based at it's just jumping out of my mind, Albany Medical Center in New York, has a very, very good mouse model of menopause they've been using for a while now, and quite a few labs are using that. And what they're trying to do is pair that with either AD mouse models or mouse models of CVID, VCID, because they're really interesting vascular impacts. And these mouse models are actually really, really very well characterized now, and they do mirror quite a lot of the sort of, at least the physiological menopause. I mean, you can't ask them about how your hot flush, but they do try to look at like skin conductance and stuff like that. But it is harder to measure some of those. Like they do try to look at measures of depression, sleep is disrupted, those sorts of things. But you know, one thing that is very difficult to characterize, and you can say this with many mouse models, is how they respond to the menopause, because a lot of women will respond differently. There's a subjective component to menopause as well. In fact, one huge thing, people talk a lot about menopause impacting sleep, when in fact it doesn't. There's many studies out there now that show that sleep architecture is not actually significant in work. Like women sleep better than men at that age, but they complain, they feel that it's a lot worse, that the quality is a lot worse. So there's a lot of subjectivity to some of this too that you cannot measure in a mouse model, but at least at the physiological level, they do have a fairly good mouse model now. But it still leaves a lot to be desired because there's been such a delay in looking at female mouse models in neuroscience as a whole. So they've got a lot of catch up to play. Even if they have that mouse model, there's still a lot to be done.
Camilla, do we have any online questions?
Yes, I have a question from Amber.
hi.
Amber asked, hi, what's your opinion about plasma-based biomarkers? Are they selectively more sensitive to AD risk in females?
That's a really good question, and one we're trying to answer now. There is some suggestion in the literature. So we are presenting some work at HAI, and we just did it, the Clinical Trials of Alzheimer's Disease Conference, suggesting that women, there's a strong association between p-tau217 in plasma and tau PET, particularly higher levels of p-tau217 in plasma is associated with much faster rates of tau PET. And that's been shown for a few years now. What we wanted to do was see if sex moderates that association. And what we found is it's a very strong moderator, so that women who have higher levels of p-tau217 have much faster rates of tau. That does not surprise us because p-tau217 is a very strong associate of high amyloid load. And we already know that women with high amyloid are not doing well. So that's what we would predict. One thing we don't necessarily see all the time in every single study, because we've looked across a few studies now. Some of our studies show high levels of p-tau217 in women, some of them don't. So it still remains to be seen. We're still not entirely sure if that's true. And also, I'm still not entirely sure what p-tau217 is actually measuring. So, you know, it's certainly not measuring tau tangles. So it's sort of, you know, which form of tau are we really? I think the thing that Gillian and I are super interested in is which part of the tau story, when in the tau story are sex differences starting to emerge. And you could say the same of amyloid too. If there was an amyloid effect, is it the soluble or the aggregated insoluble? And that's where we're starting to turn now. So it's a good question.
I think we will
Perfect.
bring formal proceedings to close.
Yeah. Thanks very much.
Fantastic talk.
Thanks everybody for coming.
Please